Cancer was long seen as a rebellion of our own cells, fought with surgery, radiation, and chemotherapy, blunt tools against a cunning enemy. But what if the real problem was a failure of the body's own defenses? What if the cure lay in awakening our immune system to fight back? In 1891, New York surgeon William Coley noticed a patient's tumor vanished after a severe infection. He wondered, could triggering the immune system destroy cancer? Coley began injecting cancer patients with bacteria, provoking fevers, and, sometimes, miraculous tumor regressions. He refined his approach into Coley's Toxins, a bacterial cocktail meant to sound the immune alarm. Results were unpredictable and sometimes dangerous, but for the first time the immune system was being harnessed against cancer. The medical establishment, wary of the risks and inconsistency, soon abandoned Coley's methods for the more controllable tools of radiation and chemotherapy. Coley's radical idea faded into obscurity, waiting for science to catch up. Yet, his experiments planted the seed for a new era, immunotherapy. The story of turning the body's own defenses against cancer had just begun. Coley's vision lingered, but the immune system's language remained a mystery. In the 1950s, scientists discovered interferon, a protein that could modulate immune responses. With recombinant DNA technology, large-scale production became possible, and hope soared that interferon could be a cancer cure. Trials showed promise in some cancers, but results were mixed and side effects harsh. Next came interleukin 2, or IL-2, a molecule that turbocharged T cells. Dr. Steven Rosenberg pioneered using IL-2 to multiply and reinfuse a patient's own T cells, sometimes achieving dramatic remissions in otherwise hopeless cases, but the treatments were brutal, with life-threatening side effects. Interferon and IL-2 proved the immune system could fight cancer, but controlling its power was another challenge. The principle was sound, the execution perilous. The quest continued. How to unleash immunity with precision. The answer would come from understanding not just how to accelerate, but how to release the brakes. For years, immunotherapy meant pushing the immune system harder, often with dangerous consequences. Then came a breakthrough. What if cancer survived not because the immune system was weak, but because it was being actively suppressed? Scientists like James Allison discovered T cells have brakes, proteins like CTLA4 and PD-1, that tumors exploit to hide. By blocking these brakes with drugs called checkpoint inhibitors, the immune system could finally see and attack cancer. Ipilimumab, the first of these drugs, led to long-term remissions in some patients with advanced melanoma. Next-generation inhibitors, like pembrolizumab and nivolumab, targeted PD-1 and proved even more effective and less toxic. These therapies transformed once-fatal cancers into manageable diseases for many. The immune system, unshackled, became a powerful weapon. The age of precision immunotherapy had arrived. Checkpoint inhibitors taught the immune system to recognize cancer, but some tumors remained invisible. Enter CAR T-cell therapy. Scientists engineer a patient's own T-cells with synthetic receptors to target cancer directly. The first major successes came in blood cancers, like leukemia and lymphoma, where engineered T-cells wiped out disease in patients with no other options. Remission rates soared, and CAR T therapies earned FDA approval. But the process is complex, expensive, and not without risks. Severe immune reactions and limited success against solid tumors remain challenges. Still, CAR T represents a leap, a living, personalized drug built from the patient's own cells. The dream of a cellular cure is now a reality for some. The challenge making it safer, cheaper, and more widely available. The frontier of engineered immunity is just opening. Today, biology and information science are merging to shape the future of immunotherapy. Artificial intelligence sifts through vast data to predict which patients will benefit from which treatments, accelerating true personalization. Scientists are engineering T-cells with logic gates, requiring multiple cancer signals to activate, reducing collateral damage. New therapies like bispecific antibodies and antibody-drug conjugates deliver precision attacks on tumors. In vivo gene editing could soon reprogram immune cells directly inside the body, making advanced therapies more accessible. Protein degraders offer new ways to eliminate cancer-causing proteins once thought untouchable. By 2030, immunotherapy will be smarter, more precise, and more integrated, guided by AI and tailored to each patient. The journey from Coley's crude toxins to AI-designed cells is a testament to the power of harnessing our own defenses. The war on cancer isn't over, but for the first time, our immune system is fighting back with intelligence and intent. The future of cancer therapy is within us.
Thanks for watching.